Hello everyone. My name is Xin Gao. I'm an assistant professor at King Abdullah University of Science and Technology in Saudi Arabia. Today, I'm going to talk about our work on development of a framework for parameter estimation of a specific biological networks, which is called gene circuit models, using structure information. So structure information here does not mean three-dimensional structures, but means the structure and the topology of the network, as I will show you later. So this is a joint work with two members of my group, Hiroyuki Kuwahara and Ming Fan, and also with Suojin Wang at Texas A&M University in the States. So first of all, I'm going to quickly go through the biological background, as most of you already know about this. We know that the genes, which I denoted as gene 1, G2, G3, their transcription can be regulated by transcription factors. When transcription factors binds to transcription factor binding sites, it can either activate or repress the transcription level of genes. And here I'm just drawing one transcription factor binding sites, and in reality there can be more than one, and our method works for multiple transcription factor binding sites as well. And the genes can be transcribed into mRNAs, I denoted as M1, M2, and M3. And mRNAs can be further translated into proteins, P1, P2, P3 here. And these proteins can serve as the transcription factors to regulate other genes' expression. So for example, in this example, protein 1 can repress gene 2, and protein 2 can repress gene 3, and protein 3 can repress gene 1. So this kind of network has a specific name called gene regulatory circuits, or gene circuits for short. The definition is that it is a functional cluster of genes that impact each other's expression through transcription factors and cis regulatory networks. And this is the model that we are going to target on in this talk. And gene circuits is a very important concept in systems biology and synthetic biology. Usually there are two problems associated with gene circuits, the forward problem and the backward problem. So the forward problem is called design problem, where we want to get a rational design of the gene circuits such that we can maximize the flux of some interesting molecules that we desire, and at the same time, we want to minimize the production of undesired byproducts. And the backward problem, usually we have a set of experimental observations, and we want to calibrate the model so that we can reproduce the observation in the best possible way. And usually this is related to either structure learning or parameter estimation of the model. And here in this talk, we assume that structure is given, we just want to estimate the parameters like the rate constants. So at one hand, the current technologies enable us to have the time series experimental data, such as time series mRNA data. And on the other hand, the parameters are very difficult to be measured directly. Therefore, the parameter estimation methods are crucially needed in order to develop dynamic models that we desire, and also in order to get a better understanding of the system level. So here I'm showing you one examples of real data where there are 4 mRNA time series data per sea urchin embryo. And you can see that we have 2 trajectories. One is red, one is blue. And the 2 trajectories are similar but not the same because of the noise embedded in this model. And also, for this model, the structure is not known. Therefore, if you want to solve this problem, you need to do structure learning and parameter estimation simultaneously. And another example is the one that I just showed you, where each gene represses its next gene. And this kind of model has a specific name called repressor. It is the first gene circuit ever that human being can design and implement. So this has been implemented in E. coli, and it can give you oscillation dynamics. And we can use the ODE to describe this model. So basically, this equation says that the speed of the change in mRNA is a function of the level of protein I-1, which is its transcription factors, minus the self-degradation rate of this mRNA. And the speed of the change for protein I depends on the translation of its own mRNA minus the self-degradation of this protein. And here we have been assuming the equilibrium thermodynamic models, and also we assume the gene expression is done through the transcription factor and cis regulatory elements. So the state of the art for parameter estimation are these three general purpose methods. The first one is SRES, which stands for Stochastic Ranking with Evolutionary Strategy. The basic idea is that once you have an optimization problem, you usually have constraints. So instead of treating those constraints as hard constraints, you can treat them as soft constraints by putting them in your objective function and put a weight before this penalty term. And that weight should not be a fixed constant, but should be dynamically changed during our optimization procedure. So they developed this evolutionary algorithm to stochastically change the weights through the optimization. But as you can see that whenever you change your weights, you get your new parameters, you need to simulate the whole ODE system, which is very time consuming. So this algorithm is very slow, and scalability is its main bottleneck. And the second method is the famous common filter. Here they have the hybrid extended common filter, which means the combination of discrete and continuous variables and with nonlinear models. And they further use moment matching to refine the results of common filter. As you know, that common filter does not require any simulation of the ODE system. So the algorithm is very fast, but it requires the prior knowledge regarding the covariance matrix of the noise, which is usually unknown or difficult to guess. And the third algorithm is called TDDM, which stands for the two-phase dynamic decoupling methods. So instead of treating those correlated ODEs all together, they treat each of them individually. So they run their optimization on each of those ODEs. And as you can see that the good thing is that they decompose the ODE system into individual ones. So their algorithm is quite fast. But on the other hand, you introduce a lot of additional noise, and the accuracy is the bottleneck for this method. So as I mentioned that all of these three are general methods that can be applied to any parameter estimation problems. And here, because of the importance of gene circuit models, we aim to develop a more tailored approach that specifically works for these gene circuit models by using the structure information, as I'm going to show you in 1 minute. So let's look at the problem setup. We are given the time series mRNA levels at discrete time points. And we also assume that protein levels at time point 0 is known, but for the rest of the time, it's unknown. And as I said that for gene circuit models, they have this specific ODE formulation where the speed of the change of mRNA is a function of all of its transcription factors. So P here does not have a footnote. So it's a vector of proteins. No matter how many transcription factors you have, you put it in this function. And minus is self-degradation. And the speed of the change in proteins, it depends on its translation minus its self-degradation. So from these two equations, if we know the mRNA levels for all the time points in this equation, then we can use this integration to estimate the protein level. And similarly, in this equation, if the protein levels for all the time points are known, then we can use this integration to estimate the mRNA level. So this gives us the basic idea of our method, where we are going to iteratively assume that the mRNA levels and protein levels are known and try to optimize the parameters. But as you can see that whenever you do this decomposition of correlated equations, you will introduce errors. And you need to have a very good refinement procedure in order to gradually refine the accuracy of your parameters. So based on this idea, we propose this framework. We call it PEDI. It's a parameter estimation framework for gene circuit models. And this is the flowchart of PEDI. The input are the ODE models of any gene circuit, and the time series mRNA level, which are discrete point mRNA levels for all the mRNAs in the system. So PEDI consists of two steps. The first step is the initial estimation of the parameters. And then there is a refinement step that refines the initial guess. So at the initial state, we just make a very brief assumption and a very wrong assumption that we will use linear assumption to link the discrete time points of mRNA together. So here, we have these squared mRNA levels, and we just use linear assumption to connect them. So if we assume this assumption is wrong, is correct, which is not true, then we will have the mRNA level for all the time points. And we can plug this into the integration to estimate the protein levels at the discrete time points. And then we further assume linear assumption to connect protein time points together. And we assume this is the true curve of protein level for all the time points, which is not true as well. And in this case, the integration does not depend on protein levels anymore, but it depends on the parameters, those thetas. So here, you need a local search algorithm, any local search algorithm, that can help you to find the best parameters under this assumption, this wrong assumption. So that's why we call our method a framework, because we can plug in any local search method in this step. So in this paper, we have been using the very simple simulated annealing for the local search step. And we assume this is the true protein curve. And we want to find the best parameters that we can find under this assumption. And suppose these circled points are the best ones that we can find. And of course, the parameters we got at the beginning are far away from the true values. So what we need to do is that we need to simulate the ODE. This is the only step in our framework that we simulate the entire ODE system. And once we have the initial guess, we simulate the ODE, and we get these circled curves. And as you can see, these curves are far away from the observed data points because our parameters are very bad at this moment. So we are going to adjust these circled curves in order to make sure that at the given discrete time points, our simulated curve exactly matches the observed data points. So for example, we drag this point down to this squared time point. And then for all the intermediate data points that we sampled, we drag them down proportionally to the distance to these two discrete time points. And in this case, we sampled 100 more time points, intermediate time points, inside each time interval in the purpose of getting a more accurate estimation of the integration. So we adjust the simulated curve to these triangle curves. And then we use these triangle curves as the true curve to estimate the protein level. And for every 10 intermediate points of mRNA, we estimate 1 protein time point. So we have 100 time points in each time interval, and here we end up with 10. And then we use these 10 protein time points to get 1 mRNA points by using the same local search algorithm. So we gradually refine our parameters, and we judge whether we reach the convergence. If not, we go back and do the refinement again until convergence. So I'm going to show you some examples that we got. And in order to evaluate our method, we used 4 measurements. The first one is the prediction error, which describes how well we can reproduce the curve of the observation data points. And the second one is running time. And the third one is how close we can get the parameters to the true parameters. And the fourth one are the power to predict the unseen data points, which means we want to use the parameters that we estimated to predict what are the future data points. And we first compare our method with all of the 3 state-of-the-art methods on this repressor later model, which I just showed. So gene 1 represses gene 2, gene 2 represses gene 3, and gene 3 represses gene 1. And the ODEs are like this for all the 3 mRNAs and proteins. And these are the unknown parameters that we want to estimate. Please note that the power here is also unknown parameters, which means this is a highly nonlinear system. And the proteins can be more than 1 in real case, but in this specific example, the regulation is only done by the protein of the previous gene. So when we compare with the 3 methods, we can see that common filter, which doesn't run any simulation, is the fastest algorithm. And PEDI has a comparable running time. And in terms of the average prediction error, best and worst prediction error, PEDI is way better than the other methods. And in terms of the accuracy of the parameters, PEDI is about 5 times more accurate than the second best method. And when we use the estimated parameters to predict the future 1 time point, 3 time points, and 5 time points, the average error of PEDI is also very good. And here, I show you the curves of the 4 methods. So this is for mRNA1, mRNA2, mRNA3. And the red dotted curve, which is hidden under the dark blue curve, is the true curve. And the red squared dots are the simulated data points by adding noise to the true curve. So the inputs are the red squared data points. And the dark blue curve is PEDI's reproduced dynamic curve. And the green one is common filter. The light blue one is TDDM, and the pink one is SRES. As you can see that only PEDI can capture this oscillation dynamics very well. And in order to compare the parameters, we draw this triangle to denote the 3 parameters associated with mRNA1. So again, the red points are the true parameters. So the closer the triangle is to this red triangle, the better the parameters are. And you can see clearly that PEDI's triangle are always much better than the other methods. And when you look at how the refinement works, at the initial guess step, this is mRNA 1, 2, and 3, we can see that PEDI gets very bad parameters. We couldn't get any oscillations at all. And starting from iteration number 2, we start to get some oscillations. And when it converges at iteration 22, we get very good oscillation. And the second experiment that we did is on these gene circuits. Here you have multiple transcription factors. So gene 2 activates gene 1, and at the same time, gene 3 represses gene 1. And gene 1 back represses gene 2. And these are the ODEs, and here are the unknown parameters. So again, we compare with those three methods, and common filter is the fastest algorithm, and PEDI is a close second. And in terms of prediction error, best parameters, and prediction power, PEDI are always the best among these four methods. And when you look at the dynamics that we produce, you can see that both PEDI and common filter can reproduce these dynamics very well. But SRES and TDDM failed to do so. And the third example I want to show is a much bigger repressor later model, which contains 7 genes. So each gene represses the next gene. And again, the ODE is generally like this. And these are the unknown parameters in this model. So you can see that the reproduced curves of gene mRNA 1, 2, 3, 4, 5, 6, and 7. And as you can see that both PEDI and common filter can give you some oscillations, but SRES and TDDM could not. And between these two oscillations, apparently PEDI's oscillation is much closer than the true oscillation. And in terms of prediction error, best parameters, and prediction power, we are way better than the other methods. And finally, I'm going to show you a real data set. So usually this kind of study is not done on real data sets because for most of the real data sets, you do not know the true parameters. So even if you get your results, there's no way that you can conclude that you are better or worse than others. And this is a real data set that contains 3 genes, GCN4, LEU3, and ILV5. All of them are very important for the amino acid biosynthesis pathways. And they form a coherent type 1 feed-forward loop. So these are the ODEs of this model, and the red squared points are the observed data, and the blue curves are the curve that is reproduced by PEDI. And you can see that PEDI can relatively well capture the trend of those observed data points. So to conclude, we have shown that the high-dimensional parameter estimation is very challenging. And we believe that the way to get scalability is to decompose the system. But keep in mind that whenever you decompose your system, you introduce additional errors. So you need to find a good refinement procedure in order to reduce your errors. And I would like to thank Gabriel for providing us the source code of hybrid extended common filter and moment matching, and Ji Guo for providing us the Yeast time series microarray data. And this work was supported by COST AEA grant and KUSCI grant. And we would like to thank ISMB ECCB organizers, committee members, and reviewers for giving us this opportunity to present our work here. And PEDI is freely available at my website. And this is the photo of most members of my group. This is Hiro, and this is Ming Fan, who are the co-authors of this paper. And finally, I would like to thank you all for your attention. I'll be more than happy to take any questions.
Thank you for a very interesting talk. So it was a bit long, but we have time for maybe 1 or 2 quick questions. Yeah.
Yes.
There are methods which are based on this direct exploitation of the data using interpolation schemes, so the generalized smoothing approach, for instance, which also exploits the structure in a very relatively similar way, not the same way, but a similar way. Did you also compare your methods to those?
No, not yet.
Another question? Yes.
I have a first question that is on the convergence. I would be curious to know if convergence is very good. You don't have, I imagine, a formal proof that conversion in any case. I imagine that you have an explanation for this. And the second question is on the Petri net.
Okay, so those are very good questions. So for the first question, that we do not have a proof of convergence. And as you see in my flow chart, we show that either we get convergence or we reach a maximum number of 100 iterations, then we stop, because we cannot guarantee the convergence. But in reality, for all the examples that we tried PEDI, we get convergence in less than 100 iterations, but we do not have any theoretical guarantee. And for the second question, that we haven't tried that yet, but thank you for pointing that out. We are doing a follow-up comparative study of a much larger scale of parameter estimation methods. So the method you mentioned and he mentioned, we probably will include in our comparative study.
So thank you very much. And,
Thank you.